So osmosis is the movement of water across a membrane, and it's really an example of facilitated diffusion. This is because it's going to use a protein helper. So there is a protein helper for osmosis. So the fact that movement of water gets its own name, osmosis, must give you a clue that the movement of water is going to be really important for cells. So let me take a second to explain why water movement is different. So let me draw a cell here. And so let's imagine that there's, again, 5 molar of something and 2 moles of something else. Not really important what it is. And then maybe there's 2 moles out here and 1 mole out here. And so notice that there is a gradient of K+, and there's also a gradient of sodium. And in both cases, the gradient is to go from out of the cell. Okay, but what I haven't emphasized yet is that the cell mostly doesn't let things move out of it. So remember, ions are a great example of something that doesn't pass through the membrane very easily. So that means that for the most part, unless the cell really specifically decides to sort of make an effort to let these out, they won't be able to leave the cell. So the cell doesn't allow K+ and Na+ to move, even though there's a gradient. So let's add some other terms here. Remember we said that we could use these hypertonic, hypotonic terms. So let me say that the environment is hypotonic to the cell, or the cell is hypertonic to the environment. Okay, so remember this means more solute inside the cell, less solute outside the cell. And so earlier we were focused on solutes, and we said, well, that would mean that solute would diffuse out of the cell, and that's what I already drew up here. But again, let me remind you that what I just said is that the cell doesn't allow K+ and Na+, even though there is a gradient. What the cell does allow is the movement of water. Cells, for whatever evolutionary reason, almost entirely and freely allow water to move in and out of it. So we need to now relate solute concentration to water concentration to know the consequences of this. So more solute means that there's actually less water, right? More solute takes up space, then there's slightly less water. If there's less solute, there is more water, right? Less solute means more room for water. So what that means is that water will diffuse into the cell from where there's more water to less water. So water will move from hypotonic conditions to hypertonic conditions. So that's what water does. Because this is the opposite of solute concentration, solutes move from where they are hypertonic to where they're hypotonic. But we use the terms about solute, so water is the opposite direction. Okay, so just kind of get comfortable with this terminology and relating it back to solutes so that you can predict the movement of water versus the movement of solutes. Again, in a living cell, you're usually going to see movement of water, not of solute. So just remember that. It's really important in the lab. That goes along with this chapter. We're going to be using living cells like potatoes. Potatoes are going to freely allow the movement of water, not the movement of salt. So if they are in hypotonic conditions, water will flow into the potato. If they are in hypertonic conditions, then water will flow out of the potato. But in no situation will sugars and salts move into the potato, because, again, the potato cells are not letting solutes move into them. So in general, the cell is hypertonic to its environment because the cell is concentrated with goodies. It's got all kinds of sugars and food and everything in it, so it tends to be hypertonic. So this means that typically water is moving into a cell, and it creates this osmotic pressure. And this helps keep cells from not shriveling. Okay. So depending on the type of cell, it will be really sensitive to its environment as far as water goes. So in an animal like us, our red blood cells are very sensitive to the concentration of solutes. And so our blood has to be very carefully maintained at a solute concentration that is isotonic to our red blood cells. This is one of the primary functions of our kidneys. They're going to make sure that the salts and sugars and everything is where it should be to make sure that it's isotonic. So in the isotonic conditions, there's just the right amount of water in the red blood cells. If the cell is in hypotonic conditions, remember, water will move from hypo to hypertonic conditions, then water will flow into the cells, and that will overblow the cells and they can actually explode. If, on the other hand, they're hyper, so remember water moves from hypo to hyper, if they're in a hypertonic condition, then water will flow out of them, and that will cause the cells to shrink and shrivel, and that's called crenation. So this is also the principles that happen in the cases of dehydration. So if you're dehydrated, you're tending towards hypertonic conditions. So you're tending towards this in dehydration. So your cells, your blood is too salty because you don't have enough water, and so water is flowing out of your cells, compounding the problem. So that would be dehydration. There's also such a thing as overhydration. This is not as common, but if somebody drinks too much water all at once, their kidneys can't keep up, and they're maybe low in electrolytes, then their blood is hypotonic. Water flows into their cells and bursts them. Probably the most famous examples of this have happened during marathons, when a runner will just chug a bunch of water, and even trying to will drink the water without really feeling thirsty because they feel like they should, but they can actually overhydrate and cause this problem. Plants have a little bit of a different relationship with water. The reason is because of their cell wall. So the cell wall prevents them from exploding. So whether a cell is in isotonic, which is up here, or hypotonic conditions, so in the case of isotonic, there's no net gain of water, so the cell is fine. In the case of hypotonic, the water will flow in and it will fill the cell, fill the cell, fill the cell, but eventually the cell will hit up against the cell wall and the cell wall pushes back. So imagine filling a balloon in a box, a water balloon in a box. You could fill the water balloon, fill the water balloon, fill the water balloon, and eventually the water balloon would hit the edges of the box. And then, if you can imagine this, the edges of the box would basically force water to go back out of the balloon. So you might keep trying to put water into the balloon, but water would keep flowing back. And that's the same thing that happens with the cell wall. Water comes in, water comes in, water comes in, but then the cell wall pushes that water back out. So again, the cell wall will push water back out. However, if cells are in hypertonic conditions, water leaves the cell, and this creates a problem. So now the water isn't pushing against the cell wall, and so now the cell doesn't have the normal turgor pressure that it should have, which is this water pushing up against the membrane. And that turgor pressure is what helps maintain the plant shape, so the cell shape, but also the actual plant shape. So as the water leaves, it loses that turgor pressure, and then in the plant, this is seen as wilting. So this is going to happen when the soil does not have water in it, right? So if I haven't watered my plant, the soil becomes hypertonic, and the plant can't draw water in. So this is a little bit of a question about osmoregulation, which is regulating water balance. So a paramecium is a kind of organism that uses a contractile vacuole, which is pictured here. So see, this is before, and this is after. It contracts. So here it's full, here it's pushed or contracted, and is empty. So a paramecium uses that because it lives in pond water. The pond water is hypotonic, so water tends to flow into the cell, and it flows into the contractile vacuole, which it then squeezes. So the question for you is, if a paramecium were to swim from a hypotonic environment to an isotonic environment, would it need to increase or decrease how much water it pumps out of the cell? So if you think about hypotonic, this would be, it's hypo. That means water would have to move in, or would be moving in, and the paramecium would have to pump it back out. So it's going to have to be pumping a lot. If it's in isotonic, the movement of water is balanced, so it would not need to pump it back out. So this would reduce the pumping that was needed. So again, I just want to reiterate as closing to this discussion of osmosis, that osmosis is so important because the cell almost always freely allows the movement of water, but solutes are almost always controlled. So even when there's a solute gradient, the solute may not move, only the water will move.